So today we're going to talk about inheritance and understanding genetics. This is going to be review for some of you, but I think for many of you, even if it is review, take the time to go through each slide. Just make sure you understand what we're getting at and the vocabulary, and it'll be a good review. So how do you describe how parents have similar traits to their offspring? Well, this was the question asked by Gregor Mendel, and you can see his picture right there. Gregor Mendel was born in 1822 in the Czech Republic. He combined math and biology, and there are many patterns in the natural world. And so we look at those patterns, and how do we understand those patterns? Well, what we need to do is we actually have to look at using mathematical computations to figure out how some of those patterns worked. And that is precisely what Mendel did. And he recognized this pattern of inheritance. Not that they didn't know that, people understood before Mendel, oh, you look more like your parents than you do some stranger on the street. However, how did that come to be was not well understood, and they hadn't been collected and analyzed in a way that made sense of a pattern. So, Mendel was very, very talented, but he also was lucky. And let's talk about his experiments for just a second. Why do I say he was lucky? Well, he happened to pick traits that followed a particular pattern that is identifiable. Now, many traits we talk about do not hold to this pattern of inheritance, and we will talk about how we figure out other types of traits. But right now we're just going to talk about dominant traits and recessive traits. Okay, so that's starting with the very basic foundation of how we understand inheritance. And this is what Mendel figured out. So Mendel took pea plants. Again, we talked about a little bit in class, but using plants, they have a couple advantages. One, you can get lots of offspring in any one given cross. So you can cross a female and a male, and then you end up with lots of offspring. So you can start becoming statistically significant. The other big advantage of using plants is that they're easy to manipulate. You can determine which plant you want to cross with which particular plant. And you can't do that just with any, with humans. That would be considered unethical. So there's many advantages to doing this. So let's look at some of the traits he picked. He analyzed seed shape, seed color, flower position, pod color, pod shape, and plant height. And oftentimes when we're doing genetic problems where we're going to ask you to identify the likelihood that we get a particular offspring, we'll use these particular traits. And the dominant trait in these, as you can see, round is dominant over wrinkled. Yellow is dominant over green, and so on. Now, with that, let's define these two words. Dominant, meaning you need only one copy of an allele, or one copy for a particular trait, in order for that trait to be expressed. And that's what a dominant trait is. So, if I have big Y, little y, I will end up with yellow seed color. I could also get yellow seed color if both the male and female parents end up giving big Y, big Y. That would be homozygous dominant. Homozygous means having the same allele, big Y, or it could be little y, but in this case I said dominant, which means it's the trait that if you just have one copy of that allele, you will end up with that trait expressed. Then there's recessive traits. Recessive traits, and there's lots of examples here, but one is green seed color. For green seed color, you need to have two copies of an allele for it to be expressed. Little y, for instance. So both parents would have to give the little y allele to their offspring for them to have a green seed color in their offspring. Okay, so what will happen when pure yellow peas are crossed with pure green peas? When I say pure, I mean they are homozygous, having both alleles being the same. Well, let's look. So if you have parent 1 having big Y, homozygous means they are both capital letters, and parent 2 being little y, you can see both alleles being lowercase letters, all of the offspring are going to be yellow. And that is because, as we reviewed in the slide before, big Y is dominant over little y, and thus, just with that one copy of big Y from parent 1, the offspring will be yellow. So we'll take this out a bit further. So if we have a tall plant and a short plant, we can cross them. So what you do is you take the pollen from one of the parents, and we'll take maybe from the tall parent, and we put it on the stigma of a flower from the short parent. We gather those seeds, and then we plant those seeds, and we make what we call an F1 generation. And you can see that here in the diagram. And in the F1 generation, all the plants are tall. What does that tell you about what's dominant? Right, the tall feature is dominant. And so that is the F1 generation, where parent 1 is crossed with parent 2. Then if you take parent, make the two parents both be the F1 generation and cross those, you get now a ratio of 75% will be tall and 25% will be short. This is the pattern that Mendel saw. And we're going to talk a little bit about how you actually then understand that using a Punnett square. So here's the Punnett square. In the case of taking two F1 generations, so here's a big Y, little y on both, they are both heterozygous. Heterozygous means hetero means different. Big Y is different than little y, and so thus is called hetero. If you cross two heterozygous plants, in this case they're heterozygous for the seed color, you will end up with a 3-to-1 ratio, or 75% to 25%. We can be expressed lots of different ways, but you have 75% chance of having a yellow offspring and a 25% chance of having a green offspring. And you can figure this out by placing the alleles down and determining what the possible offspring would be. So in Punnett square, why we use them is to get all possible combinations of a particular cross, of a particular trait. So the Punnett square review. If you have mom and dad and big sunglasses are dominant, mom gives a big B and dad gives a big B, and the only possible offspring would be a offspring with sunglasses. On the other hand, if not having sunglasses is recessive and mom and dad both are ones without sunglasses, and we know they're both little b's, all they could give is little b's. So we have one little b from mom and one little b from dad, and the offspring will 100% of the time be little b, or have no sunglasses. Okay, so let's break this one down. Mom is heterozygous and dad is heterozygous, meaning that their two alleles are different. And now we see what happens with all the possible things that could happen in a cross. So in order to use the Punnett square, you have to take that heterozygous mom, and so you put the big B on one line, you put the little b on the other line. Then dad, same thing, he's heterozygous, big B and a little b, hetero meaning different. So big B goes on one line, on one section above one of the sets of squares, and little b goes on the other, on the top of the other set of squares. And then you combine all of them. So when you combine the first allele from mom and the first allele from dad, you get big B, and so on. And then when you look at it, the genotype, you can tell I'm recording this at school. Okay, so you got a 3-to-1 ratio where for the phenotype, and that's the appearance or how the trait actually plays out, you have 3 with sunglasses to 1 without sunglasses. And for the genotype, you get a 1 to 2 to 1 ratio. 1 big B, 2 big B to 1 little b. So that is how you figure that out. Okay, so what is independent assortment? It's that alleles separate independently during the formation of gametes. And you remember that if any of these words are vocabulary words for you, you should be writing down the word and then making sure you go and get the definition of it. For instance, gametes are sex cells. In animals, those would be egg or sperm. In plants, those would be pollen, or the bottom of the stigma, which is the place where a female part of a plant is. And then when they're making those gametes, you have alleles. And alleles, if that's a vocabulary word, remember, write down allele and then and then work with that. Alleles are one variation of a gene. So, for instance, the big Y or the little y, those are both considered alleles. And when those, so if you have alleles for one gene and then alleles for a different gene, they don't get hooked up together. They will be separately sorted. So that's how you could have blue eyes and blonde hair, or blue eyes and black hair. And that's just kind of a simple example, and it doesn't exactly work genetically, but we'll talk more about that later. But that's what we're going to be getting at later as we progress into dihybrid crosses. So we will be moving on with more inheritance very soon.